Hello, I'm MeiLi Papa presenting my thesis titled Pathogen Classification using AI-enabled hyperspectral microscopy to detect biological variations. Every year in the United States, 1 in 6 people get sick from a foodborne illness. Conventional detection methods take a long time and require trained personnel. One biological variation I looked into is viability, and stress pathogens can enter a viable but non-culturable state that can be induced by environmental stressors. VB&C bacteria can bypass conventional culture-based detection methods because of their non-culturability. The other biological variation I looked into was surface structures, specifically serivars, which are groups within a bacterial species. As you can see, all of these images are of Salmonella, but they are all actually different Salmonella serivars, so they look spatially similar, but on the microscopic level, their surface structures are different. So for the VB&C induction methods, I started with inducing the VB&C state for E. coli K12 with peracetic acid and hydrogen peroxide. I then confirmed their viability and non-culturability using a live dead staining kit and plate counting. After that, I went on to collect hyperspectral microscopic images of the E. coli, VB&C, and normal cells. And this HMI data collects 303 bands of information from the visible to the near infrared range. Then, after all the data was collected, I then pre-processed it by selecting 3 out of the 303 bands, which are the most important bands, to create pseudo RGB images that were then augmented and then sent into EfficientNet v2, which is a deep learning model that classifies between the normal and the VB&C cell images. For the results, the standard color images were used with EfficientNet and had an overall accuracy of 83%. And conversely, using the pseudo-RGB images from the hyperspectral data had an increased accuracy of 97%, and this study laid a solid proof of concept foundation for using an AI with hyperspectral microscopy for pathogen classification. So with the same sample prep as before, 500 hyperspectral images of 5 Salmonella serivars were collected. Spectral data was pre-processed and used with machine learning models. Then on the bottom, image data was pre-processed and used with EfficientNet, and the outputs were combined together for a fused classification of Salmonella serivars. So this image shows the confusion matrix for the serivar classification using the multimodal fusion, which uses both spectral and spatial data. The rows represent the true label, and the columns represent what the model predicted. There is an overall accuracy of 83% for classifying the serivars, and these results show that data-driven pre-processing and multimodal fusion allowed for improved Salmonella serivar classification.